Hello, I just wanted to welcome you to Biology 127. I'm really excited for our year this year. This is going to be a new adventure, and let's get started. So this year, here's my contact information, ljcook@northcentral.edu, and in class, I will be taking attendance, prayer, and then we will go through the introduction and syllabus, office hours, and answer any questions about lab in the lecture overview. So we will cover that in class as needed or via online. So the class or the syllabus will be on Canvas or Moodle. The office hours, I hold office hours in lab and office and by appointment. And then I usually give a mini lecture overview. We covered the syllabus review with any questions that we had. A little bit about me. I attended Central Michigan University in Mount Pleasant, Michigan. I am from Michigan. And I also attended Eastern and Western Michigan universities. Eastern Michigan is close by Detroit, and Western Michigan University, I attended in Kalamazoo for my PhD in science education. Eastern, I did field research, and for my dissertation, I did classroom research. Some of the places that I have taught over the years are here. North Hills Classical Academy is a K through 12 school. I taught at Davenport for many years, and also at NCU. Couple things about this class. It's really helpful to go through some of the study tips. I gave a handout potentially in the notes section, but here are a few things. These are tools that will be really helpful for when you go through this class. The textbook, I have included that information in the syllabus, and then we have study groups and that I allow to start organically, and then I may ask in the future for some guidance. And then a couple things, we'll use flashcards, and we'll kind of talk about this a little bit in class as well and through the discussion boards as needed. But a couple key things is you start studying for exams early and you take great notes. Learning outcomes. Learning outcomes are the study guide for my class, and this is just a tool. Learning outcomes will help focus you in how to study. The other thing is you may need to study beyond these to get the full amount of information that you need to prepare for the assessments. And it will help because it will coincide with the test material, or the lab material, I mean. When we are looking at different ideas of science, I kind of start out with this, like, what do you think this is? This is pollen. This is pollen underneath a microscope. What do you think this is then? This is fungus on a tree. What is this? This is the eye of an ant. Where would you find this organism? This is a bed bug. What are these used for? The white tufts are inner ear hair cells that collect and that allow sound to flow over them. Can this be bad? Yes, this is what cancer can look like when you add a fluorescent dye to it, and you see that yes, it can be bad. Biology, we're going to look at our notes packet, so you'll want to have your notes packet out. In your notes packet, you will see that there are spaces where I've allowed you to write in different notes, and I will help you kind of focus some of that. The red words are the words that you want to stay focused in as you continue to learn how to take notes. And the first one, looking at Biology is the study of life. Bio meaning life, and ology is the study of. So what is life? Living and non-living. And we may classify that into, we may have done a sort like this before, where we had to classify living versus non-living items. And in this sort, we then began to understand what is biotic or abiotic. Abiotic factors can be things like weather, resources, pH, salinity, temperature. Biotic can be things like pathogens or bacteria. They are a quality that impacts the things around us in a way where with other organisms, right? They are living. So one of the first things that we will look at are the characteristics of life and what are the living things. And you'll see two red spaces here, or red words that you'll put into the spaces. And we'll go through, this is the list. And how you study a list is a couple ways. First, you want to know what's in the list. And knowing will be like, sometimes you sort them and you create like a mnemonic device. Other times you want to know what's not on the list. That's a different way of studying. You have to know what's on the list to know what's not on the list. So how do you study a list is one of the factors or a study tip. And we will go through different study tips as we progress through the lecture notes. So levels of biological organization is one of the characteristics of life. So atom is that living or non-living? And it is non. And atoms build or combine to make molecules. Molecules combine to make a cell. Cell is the smallest unit of life. That is the definition of a cell. Cells, multiple cells make up a tissue. Different tissues make up organs. Multiple organs make up organ systems, and organ systems make up organisms. Now, a common misconception is that oftentimes when we start talking about organ systems, we often think that it's just animals, but rather it's plant and animal. So we'll see this on the slide right over here. And in this slide, we can see that we have a variety of examples showing the level of diversity, but the level of organization. And as we continue to look more, we have population. A population is the same type or species living in a given area versus a community, where all populations of all species are in a given area. And then we have ecosystems. And in Minneapolis, or in Minnesota and Michigan, we have different systems that are provided, or that are very diverse, that we have a variety of ecosystems. So a prairie ecosystem versus an aquatic lake ecosystem. And then we have a biosphere. Here's a picture showing that. Another characteristic of life is the energy, that characteristic of life that requires materials and energy. Energy is the capacity to do work, and the metabolism are all, you can underline the word all, chemical reactions. So we have things that capture energy and release energy. So food molecules will help release energy as we eat, whereas we eat or consume energy. So we have energy that is used by living organisms. We call those captured by sunlight, are photosynthesizers that use light to make food. And so this is a level of organization. We want to stay organized when we have energy, and this can yield motion. So the flow of energy is that producers make their own food. So an example of that we can see in this chart would be maybe grass. We have nutrients, and this is important for growth, and consumers are going to eat off the other organisms. An example of that would be maybe decomposers. Energy is not cycled, and it is considered one way. So tertiary consumers may consume secondary consumers and primary consumers. And so we can see quickly how bio remediation is impactful in this situation. Here's a picture of a hawk and the hawk babies. Living things reproduce. Reproduction is how parents produce offspring. Oftentimes I use my own family as an example of that, to talk even about genetics and how genetically or phenotypically my kids are very different than each other, and they have the same dad. And so in reproduction, we need life to make life, and DNA is a genetic or hereditary information, and in genes and genes and DNA molecules will allow for organisms to reproduce, and we also want to create diversity so we don't look like our siblings in every case. Furthermore, we have inheritance of the transmission of that DNA, and we have two main types of cells, unicellular and multicellular. Now we also have plant and animal cells, but in this case we're talking about unicellular organisms, and they reproduce asexually, meaning that they divide themselves. So they reproduce within the same cell, or they multiply the same genetic information and then divide into two cells. Multicellular will reproduce sexually, and each parent contributes half of the genetic information. When we look at genetic stimuli, we can see that we have a positive response or a negative response. And so a positive response is that they're going to move towards a stimulus. And then we have a negative response where the movement is moving away from the stimulus. And this can also lead to components of behavior. So when I tell my kids to not touch the stove because it's hot, and if somebody comes up and they touch the stove, a negative response is moving away from the stimulus, so they move their hand away. What is an example of a positive response? A positive response would be like a flower turning its head to absorb to the sun, right? So it's going to, like a sunflower, it's going to have a positive response towards the sun, and it's going to move towards the stimulus or the sun. Another characteristic of life is homeostasis. And when we look at homeostasis, we see this in a variety of ways. One of the ways is like our heart rate. We want all of our organ systems functioning well. And when we see this, we see that we have a control center, and things can speed up and decrease our heart rate. And our body is constantly compensating for this through a variety of systems. Another characteristic of life is growth and development. Growth is an increase in cell number, and development is by which the first cell of a new individual becomes a multicellular adult. The difference of this could be humans versus insects, and insects go through metamorphosis. And I don't want to leave out our plants. So plants also go through this process of growing and developing. And then we have things called adaptations to the environment, where we see evolution changing over time. This is how we get new variations within a species. So we will look at this more in depth in biology too, if that's your next class. And we see how evolution is that individuals survive so they can reproduce, right? The strong survive. So when we look at these adaptations, we see that they will maybe change or their form or structure so they can actually survive in their environment and fit into their changed environment. And the example that you see on this picture are the colored moths. Again, here's a list of characteristics of life, and knowing what are these characteristics and what are examples of these characteristics. So here is where we see and where we will talk about biodiversity. So with biodiversity, we can see that we have different taxonomic kingdoms of life. And here we have cell types, And we can organize them in a couple ways. We can organize them by the cell type of eukaryotes versus prokaryotes. And in that organization, we can see that eukaryotic cells have membrane-bound organelles, and they're unicellular and multicellular, which is what we also have already talked about. Another feature are being autotrophs versus heterotrophs. Autotrophs obtain food molecules. Heterotrophs absorb or ingest it. We are considered heterotrophs. And then we have prokaryotic cells, which lack membrane-bound organelles. So when we look at these cell types and biodiversity, we are then going to see that we need to organize this information and classify this information in an orderly way. And somebody who has done this would be our father of taxonomy, Carlos Linnaeus. And a taxon is a grouping of these organisms. So each organism or species is given a unique name, and they have these shared traits. And so how we organize them is domain, kingdom, phylum, class, order, family, genus, species. And we use the idea of using Latin to organize these groups, and each of these levels are a group. And in this group, we have different characteristics that we define into a dichotomous key. So let's go into the binomial nomenclature. We have a genus and a species or a specific epithet. And in this case, it's Homo sapiens. The second part of the name is sapiens. That is a species epithet. The first name, or the first part of it, is genus, and this is where we have a group of species that share that same unique set of traits. So we can see this example right here. We also show authorship. And we also see that you can have different authors, or the original author or the author of the combination. And then we have a full scientific name of the species. A couple things you'll notice here is when you underline this, you can italicize it or you underline it. And you underline it not with a single line, but you actually underline it in a line right here. We can see here that we have common names in binomial nomenclature. And in the binomial nomenclature, we know that they have they are italicized or they are underlined. If they were written or handwritten, we do not use one solid line under the binomial nomenclature name. We actually separate it into two lines. And so you can see that here a pea plant is Pisum sativam. Another common one that we also see is a frog, Rana hexadactyla, is another example of a scientific or binomial nomenclature of a frog. How do we organize these ideas? Well, you can see we have screws and nuts and nails, and how we organize them are by general to specific characteristics using a dichotomous key. So here we have a dichotomous key of candy, and when we are looking at a dichotomous key, we have general ideas at the top, or number 1 or 2, and the more we go through the list, it becomes more specific. And by doing that, we can then get down to the genus or the genus and the species. And that is how we classify organisms. And so what a lot of scientists continue to do is, as they classify organisms, they are organizing them into a 3-domain, 6-kingdom system. Now, when I went through school, the textbook was the instructor actually taught from the Whitaker system from 1969, and it took a long time to integrate that into the textbooks. How I teach it is the WUSI. Your textbook is very similar to the WUSI. And what we're finding out now is, as we go through this classification system, we have things that are going to be very similar and different based on the phenotype, the physical characteristics that we've used to classify organisms, versus the genotype, which is the genetic information that we've used to classify the organisms. And so this is where the three domain and six kingdom system is getting challenged a little bit more, where we will see more and more genetic information driving this classification system scheme. Right now, though, we are going to stick with the three domains and six kingdoms. And here's a great chart to help us understand and fill out our study tools, maybe, of the different characteristics of the classification of these living things. You are responsible for knowing the three domains in the six kingdoms and the kingdom phylum, class, order, family, genus, species, sequencing. Not specific like off this biological classification chart, just the sequence of that, what I just said. This is what we're looking at with the tick, and we can see how when we go through this process, we are actually going through a classification dichotomous key for this particular species if we were to discover it or even find it out in the field. Now, this concludes a lot of our conversation about classification until we get into the second semester of biology, and then we talk a lot more about this in depth. So our next topic of study is that scientific methodology. And in the methods of science, there are three main tools that we use or do, you might say. And in this process, we make observations, we make models, and we make experiments. And in here, we want to find new relationships. We want to find things that represent and explain things. And then we have structures and functions that we can maybe observe. When we do this, we use different reasoning and deductive reasoning versus inductive reasoning. Inductive reasoning is drawing a conclusion based on the observation. Deductive reasoning is using a general idea to make a conclusion about a specific case. So here is an example of that. I will let you use this link of what we will look at in class as well. Additionally, we have a ladder of inference, and when I use this in the class, we go through the tricky tracks. And what we do is we make observations, and the easiest way for students to understand what this looks like is what are some inferences. And inferences, this is in your notes. And inference is like a story, and that one way that I teach this is through using animal tracks. So we have three observations, and then what are some observations about this? Write them down. One observation is there is a pink and a green type of track. Another observation is they are moving at an angle. We do not know the direction. So an inference would be they are moving towards each other. But that's the story of our observation. So if we look at the next slide, look at and make three more observations. An observation that we can say is during a time, the pink tracks encountered the green tracks. How would I say that, and how do I know that? It's because one of the things that I'm looking for is they're not overlapping. So I can't say the green came early or later. Maybe I can, but then I would probably see an overlap. So because there's no overlap of the tracks, I'm saying that they actually came at the same time. We could also say that they're pointed in different directions. They're not linear anymore. The tracks are not. And we could make a story, or the inference is they got into a scuffle. And we can keep going, and here is the last one. So what are 3 observations versus 3 inferences? An observation would be that the tracks are not returning from where they came from. They're continuing on the journey. The green tracks. We can also say that the pink tracks are no longer around it. The story is maybe the green track organism ate the pink track organism. We don't know, but we can create a story. The other thing is we are very careful about our objectivity. So objectivity versus subjectivity. The influence of my understanding or my thought pattern is coming from my own ideas of life and my own stories, right? That's subjectivity. In science, we want to be objective. One of the ways that we do that is looking at data and evidence, and that the evidence is relevant or relating to a subject, and that the data is valid. How we look at data validity is it precise and accurate? You can have precise data and not have it be, you can just have accurate data, but we want both accuracy and precision. Is there one method of science? In a lot of textbooks and a lot of misconceptions, there is an order or a process. This would be a misconception, I would say, in science, because just because we got results doesn't mean that that's our conclusion. We could still keep running data. We can still keep trying to find information. So I always challenge this thinking that we have a linear pathway of the scientific method. So in that idea of scientists use experimentations, is science linear? No, we know that. It can be very complex quickly. And so when we have experimentation, we have observations. And from those observations, we create a hypothesis. We formulate a possible explanation for a natural event. And this needs to be testable and use measurable variables. So one way we do this is through the if-then statement. Then we go through and design an experiment. And we have this experimental design to use experimental variables, response variables, and control variables. The independent variable versus dependent variable. Dependent can be observed or measured. Control is kept the same for all conditions. And then we have independent variables where the variable can be controlled. I like this picture on slide 58. Looking at here, the independent, dependent versus controlled. You will be responsible for knowing and discerning this from a research project. Furthermore, the data needs to be observable and measurable, and the data needs to be analyzed appropriately and accurately using statistical analyses that are for that particular data. So we can collect measurements as quantitative data, or we can collect observations as qualitative data. And when we do both of them, we do need to account for bias, but we also do need to code the observations. And we go through a whole coding process when we do qualitative data. Furthermore, when we look at results, we want to make sure that these results can be duplicated by other researchers through a process of replication. This is looking for the validity, and if we have internal, external, or if we have threats to our validity. Furthermore, we want to have controlled conditions and that our results can actually be replicated. This is a big thing in science. This has just happened recently where a research lab studying dementia was outed by another research lab that couldn't replicate their data that they've published. And unfortunately, it came out that this lab that created all these publications about dementia research actually fabricated their results. This is difficult. This is not good. This throws a lot of errors. This threatens the integrity of science. And when we do that, we are not accounting for anomalous data. It's very important to consider this and to consider all of your data and explain that in your conclusions and report with integrity and publish with integrity. One of the ways that we do that is through a peer review process. I have been on several journal articles where I've gone through this peer review process. It can be really intense for authors just as well as peer reviewers, and it brings authenticity, integrity, and objectivity into this field of publishing our research. Now, what has happened in the last few years since COVID, because of the influx of COVID research, they eliminated the process of peer review. Now, how is that strengthening our ability to report and publish our results? I don't believe, this is my personal opinion, I don't believe that's strengthening our field of research and science when we take that out. We actually weaken this process. So this is a science journal. You submit your article, it goes through review. You either get rejected or you can go under another review where they reassess it. They can reject it again or they can accept it, and then it goes through publication and then to be printed again, production and then publication. Open access, the term for that, we use an open access book sometimes, and this is where a lot of people provide input and eyes on a project, so that way it can be distributed free and online without access barriers. When we're talking about papers that have gone through the peer review process and been retracted, this is where we have an error. This is where we have errors in our research, how we published it, was it biased, was it not biased, did we account for everything? And this happens. This is very detrimental to your scientific endeavors, your ability to get grants even, and this is not a good thing in your scientific field of research. When we look at theories versus laws, theory is a testable explanation for a broad segment of natural phenomenon, whereas a scientific law, after a series of experiments in a concise statement, describes a fundamental relationship of nature. The law simply states the finding but does not explain the behavior. And when we are looking at this, there's two websites that are helpful. The Aristotle de Linnaeus website, and then we have this group of the International Commission on Zoological Nomenclature that actually go through and approve scientific names for organisms, which is kind of a fun resource to know about. Thank you for listening.